So this component now we're to discuss the protein structure determining enzyme specificity. So we know the two different modes of enzyme specificity. We're looking at induced fit, right? And we're also looking at lock and key. Right, so we're looking at the fact that the structure of the proteins are important to the enzyme specificity, meaning that if we have the structure of, let's say, protein A, the enzyme will be conducive to protein A, but not conducive to protein B. So we're looking at the fact that the tertiary structure of proteins or general structure of proteins can determine which enzymes would actually react with it for the mode of action, right? And we're generally looking at the structure of the proteins, right? And their adaptiveness, right, with the or the complementary shape of the proteins with the active site. Right, so we speak about the induced fit. We're saying that the induced fit will basically be the enzyme changing or wrapping itself around or changing slightly the configuration of its active site to bind to specific proteins, and this allows for one enzyme to work on multiple proteins. And then when we look at the lock and key hypothesis, we're looking at the fact that only one true protein structure can react with only one single enzyme. So we could be looking at that as well, right? So what we're basically going to be doing is discussing how, when protein structures change, how does that affect enzymes, right? Specificity. Because we're saying that enzymes are specific to different protein structures. So if we change the protein structure, how will that affect the mode of action of the enzyme? That is what we're asking here, right? And those are two key criteria used to answer that question. Just generally 15 marks. That is actually a module one question, my bad. But let's look at this. Right, describe the rules of DNA and RNA in protein synthesis. We know that generally DNA is the molecular workbook, the molecular the area in which we actually get the information from, right? So we know that within the rule, within protein synthesis, we have transcription. And that is really deriving mRNA, getting mRNA from DNA. Right, so DNA contains the main genetic information from which we get the mRNA, which now the mRNA would now combine to the ribosomal complex. So the ribosome complex, which contains the alpha and the beta component, right, or it contains a complex containing rRNA. Containing rRNA, right? And then when that complex is formed now, right, as the ribosomal complex, right, shifts along the mRNA, tRNA, right, will transport or carry the complementary amino acids. Amino acids. Alright, let me look at the screen and fix any issues now. Alright, I don't know why I look at the keyboard when I'm typing. Alright, so we're basically speaking about the role of DNA and stuff like that. Alright, so generally we speak about the role of DNA as the main information reservoir that we get the entire sequence from, right? We're going to speak about the central dogma of genetics here, that DNA, right, codes for the RNA and the RNA codes for the protein polypeptide chain, right? So we're looking at that. So the DNA is the area in which we get the information, right? That is how we get mRNA. mRNA is generally the stored information transcription of the sequence that we get from the DNA, and then the rRNA is what combines with other lesser ribosomal components in order to actually produce the ribosome complex for the mRNA to read through the ribosome complex, and the tRNA will be carrying amino acids from the cytosol towards the complexes to actually create the polypeptide chains. So you're speaking about describing the rules of DNA and the different types of RNA. within protein synthesis. Shouldn't be a problem, right? Now for genetic engineering, right? Genetic engineering and gene therapy are two close related technologies with promising potential for improving human health. Outline the principle of genetic engineering, including a brief description of the steps, right? So the general principles of genetic engineering speaks to the creation of recombinant DNA using restriction enzymes and ligation enzymes. Right? And we know what these enzymes do, right? General structure now, right? First, we identify the sequence, right? Identify. Oh, I've identified. All right. So first, we identify the sequence, and that can be generally done using restriction enzymes, right? So the identification and cutting the sequence, right, would be using restriction enzyme, right? So the restriction enzymes will actually help to identify the sequence we want because it cuts at specific sticky ends. Please remember that. So we're looking at what when we have BAMH1, right? We're looking at HIND3 and stuff like that. You also have HE2, I think. Please fact check that, right? HE1 or HE2, right? So you're looking at stuff like that. You have HIND3, right? So all of these stuff, right? So they'll identify specific codons, right? That it will attach to different palindrome sequences that it will attach to produce sticky ends. And the syllabus specifically speaks to them being called spoken to as molecular scissors. Alright, so the syllabus specifically speaks to them being referred to as molecular scissors, right? So we have that there, right? After that, now, after we have isolated the genetic material, right? Generally, that's from that's actually after we extract the plasmid, right? Identify the information, right? And then we restrict it, right? What's gonna happen then there, right? We're gonna take our desired sequence. So take the desired sequence. It's really early. I don't know why all the spellings are going off. Desired sequence. We take the desired sequence, right? And we add to the plasmid. Right? And then we use ligase, right? In order to act as the molecular glue. Right? To now completely bind the new sequence to the old sequence. Right? And that's now the creation of our recombinant DNA. Right? That's really the creation of our recombinant DNA there. Right? What happens after that now? Right? We're going to be in order to check, because some persons will include this check, in order to check that the recombinant DNA has been produced, we use gel electrophoresis. Electrophoresis, right? And in order to check that the recombinant DNA has been formed, then we have bacteria transformation, or right transformation, right? Or we just have use of vector, suitable vector, by the way. Right, so back to your transformation, what does that speak to? It speaks to using calcium chloride solutions, right, in order to weaken the cell wall so we can directly implant our recombinant DNA into the bacteria. Or we can use a suitable vector for the vector itself to enter the bacteria, implanting our recombinant plasmid. Lovely. Right, and then after that, now we're going to have growth culture. We're gonna have selective growth culture. Usually contain, why is it selective? We're selecting for the organism that we just modified. So usually it has some antibiotic component. So when we place antibiotics onto our culture plate, it kills off everything else that is not our genetically modified organism. Or use selective growth media, right, that actually allows for the proliferation of the organism that we want and that we modified, right, while it allows for all the other organisms that have not been modified to die off, right? And after that, we do subcultures in order to grow the organism, right? In its in a pure form, right? So that's generally what we do, right? It's just to isolate the the GMO, all right? Lovely. So we have that entire question done. All right, 5 marks there. No, despite the promising potential, right, one of the arguments sometimes raised against gene therapy is that it's technically too dangerous. With reference to cystic fibrosis, discuss this. Now we know that cystic fibrosis is a genetic disease that really affects the production of mucus, right, especially within the alveolar membrane, within the lungs, within the respiratory tract, generally, right? So we're looking at the fact now that since it's a genetic disease, some approaches would be using gene therapy, right? Now, gene therapy can be dangerous in mainly two ways. In which, right, when we use gene therapy, right, to implement our disease delivery mechanisms for gene therapy at all the time, right, really wasn't specifically targeted. So since it's not really specifically targeted, or it's a little bit difficult to target specific areas which are affected by cystic fibrosis, right? Some of that genetic information may go and be absorbed into other parts of the body or other parts of the respiratory tract, leading to multiple genetic issues, right, within um the over the overburden of genetic material within those cells, right? So that increase in genetic material within the cells that are not affected may cause an issue, a probable issue, right? Also, with the certain vectors being used, right, certain vectors may cause different types of infections within these patients, right? So depending on the vector, we may have potential issues, right, with the vectors actually ramping up their virulent or pathogenic components, right, and actually causing some amount of secondary diseases. That is two. And three, we could potentially have allergic responses to the vectors used, right? So allergens may be produced that will result in allergic responses, right, within the patient, right? So those were three there, right? And then we head into the module 3 component of section B of 2017.